Welcome everyone. Here we are at week number 4. We are almost halfway through the term. So let's take a look at what we did last week and see what we're going to be doing this week. Okay, so last week you looked at diffusion of iodine molecules into potato cubes to see how that process worked. You also did an experiment with osmosis where you looked at different concentrations of sugar and how they affected the fluid volume inside the dialysis tubing, which acts like a plasma membrane. So that should be interesting for me to grade to see how you did on that. We also studied about cells, the 2 different types of cells, prokaryotic, also called bacterial cells, and eukaryotic cells, which includes our cells, and viruses. And last week I talked a bit about SARS-CoV-2, which is the coronavirus that has reached pandemic proportions all across the globe, and its resulting disease, COVID-19. So that was a pretty busy week. You also submitted your milestone 1 for your final project, so I will be grading those too this week. So what is happening this week? This week we are going to learn about cellular respiration and photosynthesis. And mainly the experiment is about cellular respiration, but I'll cover both of these. So cellular respiration is a process that all aerobic organisms go through to produce ATP, and ATP stands for adenosine triphosphate, which is the most important energy molecule in all organisms. It's the energy molecule that provides energy to all the enzymes in cells for them to do work. And the enzymes are important because they build and break down all the molecules in your cells. So they have to have an energy source, and that's ATP. Well, where do we get ATP from but from food? So all of us who are aerobic, and that includes human beings, we have to have oxygen in order to survive. All of us who use oxygen break down our food through the process that we call cellular respiration. Well, how does that work? The food, the most basic food is glucose, which all organisms can use. So food is broken down through the use of oxygen, and the waste product is what we breathe out, which is carbon dioxide. So carbon dioxide is the waste product produced through cellular respiration. All right, cellular respiration happens in all of our cells and everything else that is aerobic, every other organism on the planet that is aerobic. Photosynthesis is sort of the opposite reaction, but it's also considered a complementary reaction. So photosynthesis is carried out by organisms that can make their own food, the autotrophs. Auto means self and troph means feeding. So all of these organisms can make their own food. Wouldn't that be cool? If all you had to do is go stand out in the sun and make your own food, you wouldn't have to eat anything, would you? That would make a cool science fiction story. So think about it. So photosynthesis. Photosynthesis is the process where glucose is produced through the use of sunlight energy. So sunlight provides the energy source to take a bunch of carbon dioxide molecules and mush them together, make them form chemical bonds to produce glucose. And glucose is the food source that these photosynthesizers are producing. Now, when they do this, oxygen is made as a waste product. They give off oxygen as waste. So, of course, they are really important for us because they put oxygen into our atmosphere that we breathe in to make ATP from our food. So I said that these are sort of opposite reactions because you can see with cellular respiration, you break down glucose and produce carbon dioxide, and with photosynthesis, you make glucose from carbon dioxide. But they're also complementary. So any of the plants, any of the other photosynthesizers like photosynthetic bacteria do photosynthesis where they're making their own glucose, but they also do cellular respiration because then they have to break down that glucose to make all the ATP molecules that they need to run their cells. So just realize that. They're opposite but also complementary. All right, you've got an experiment to do this week with respirometers. So respirometers measure the amount of cellular respiration that is taking place. In what? In peas. So you will germinate part of the peas that you got in your kit, and this takes a couple of days to do. You have to activate them. They're dried peas, and so you have to activate them and make them come back alive, right? So you're using part of the peas to germinate, and the other part you will not germinate them. You will leave them in the dried form, and you will also use some beads in the third respirometer that you are going to set up. So this germination process takes a few days. So the first day you soak your part of your peas overnight. You must use unchlorinated water. So distilled water is fine, bottled water is fine, not tap water, not straight out of the tap. You're going to kill your peas if you do that. So make sure you use unchlorinated water. Most of us have bottled water in our houses. Filtered water would also work if you have one of those filters on your faucet. So soak your peas overnight. And then the second day, you're going to put them on wet paper towels and cover them up and let them sit overnight. That's a couple of days right there. Then you're going to set up the respirometers, can't say the word, and conduct the experiment. You are going to be using potassium hydroxide. It is very caustic. Be extremely careful with this stuff. Use all of your safety equipment. Like is explained in the lab, be very careful with this. The potassium hydroxide gobbles up the carbon dioxide that's released as a waste product of cellular respiration. And by doing that, that causes the liquid that's in the end of the respirometer to move to the left toward the respirometer because you're using up the oxygen that's in the air. Okay, read through this very carefully in your lab. But the potassium hydroxide is important because it uses up and precipitates the carbon dioxide that's made as a waste product through cellular respiration. All right, a reminder, make sure you use your lab report forms. Your labs have to be turned in on these lab report forms because so much information is there. You have to answer questions, you have to fill in tables, you have to make graphs, etc., etc. So use your lab report forms and make sure you combine everything into this lab report form. I don't need 5 different documents that I have to look at. They all should be together in that lab report. And there are instructions in that lab report that tell you to do this. I will also include in the announcements this week how to use parafilm because I'm sure you have never used it before and you have to use it in this lab. It is a very, very cool substance. It is like stretchy wax paper. It is so awesome. You use it all the time in labs, but you probably haven't used this. So watch my very short videos, just a few seconds long, on how to use parafilm when you're setting up your respirometers. And as always, if you need anything this week, please email me, m.sigmon@snhu.edu. I am here to help you. So let me know if you need any help. I am here. So call on me. Have a great week.